Animal cells. There are 5 parts to know in an animal cell. We call these organelles. The nucleus controls the cell and contains your DNA, genes, and chromosomes. The cytoplasm is the jelly-like substance where chemical reactions happen. The cell membrane controls what goes in and out of the cell, a bit like a bouncer on a door. Mitochondria release energy through aerobic respiration. So the more active a cell is, the more mitochondria it has. So think of something like a sperm cell that has to swim about. It needs lots of mitochondria to provide energy. And finally, ribosomes. Ribosomes make proteins. So think about the keratin in your nails and also your hair. For the exam, you need to be able to label all 5 and say what each one does. Don't fall into the trap of saying the nucleus makes proteins. That's the ribosome's job, for example. I see lots of students mixing up the role of the different organelles. A plant cell has everything that an animal cell has, plus 3 extras. Firstly, a cellulose cell wall for support. Think about something like celery that's really quite tough. Well, that's largely due to the cellulose cell wall. Chloroplasts, where photosynthesis happens, they're full of a pigment called chlorophyll, which is why they're green. And finally, a permanent vacuole filled with cell sap that keeps the cell turgid. When we think of turgid, we're thinking of something tough and a little bit swollen. It's an easy mark to lose. Don't say chloroplasts are in every plant cell. For example, root hair cells don't have any chloroplasts because they're underground where it's dark and they don't photosynthesize. There's no light there. Examiners love asking that question. Something else I see is that students tend to think that root hair cells are like the hairs on our heads. And they sometimes think that root hair cells are in animals. They're not. They are in roots of a plant. And they have these things that look like hairs. They're long and thin to absorb as much water from the soil as possible. Now we move on to the light microscope. A light microscope shines light up through a thin specimen. So our specimen might be something like a skin cell, or a cheek cell, or a plant leaf cell. You'll remember having seen a demonstration or done an investigation where you've taken a really thin slice of onion and put it under the microscope. Well, that specimen, that onion, was really thin so that light could get through it. In a microscope, you have 2 lenses that do the work. The eyepiece lens at the top, which is where you put your eye and look down, and the objective lens, and that's the one that you can swing carefully into place over the specimen. So because there are 2 lenses, we need to think about the total magnification using the eyepiece times the objective lens. So a 10 times eyepiece with a 40 times objective lens gives you 400 times magnification. So just remember that when you are doing the calculation for magnification using a microscope, there are 2 lenses, and therefore you have to think about the eyepiece lens and you multiply it by the objective lens to give you the total magnification. Light microscopes go up to about 2,000 times magnification. So that's quite big. However, the magnification is not powerful enough to see really tiny objects. And that's where we use electron microscopes. They go up to 2 million times magnification and with a much higher resolution. And that resolution allows you to see an object more clearly. But the problem with electron microscopes, apart from being so expensive and generally very big, is that they can't show living cells. The cell has to be dead first. So that's a quick comparison that the examiner often asks for. Cell division, and we look at the first, which is mitosis. Mitosis produces two genetically identical daughter cells. Before it starts, in interphase, the cell copies all of its DNA, and it starts with copying because it's going to end up with two copies of the original cell, so each of those cells needs to have one of its own copy. So you double the amount so that when it splits into those two daughter cells, each daughter cell has its own full copy of chromosomes. Then there are 4 stages: prophase, metaphase, anaphase, and telophase. And the mnemonic for that is IPMAT, IPMAT. After IPMAT, you have cytokinesis, where you actually have the splitting of the cytoplasm. Stem cells are special because they can differentiate. That basically means they can turn into any specialized cell. In the embryo, they can become anything. So an embryonic stem cell could become a liver cell, could become a nerve cell, for example. And that's really important because as an embryo or a fetus grows, it needs to develop into different body parts, and those different body parts have different functions. So it's really important that embryonic stem cells can become any other type of cell. Adults also have stem cells, but they are much more limited. For example, bone marrow. The stem cells in bone marrow can go on to become different types of blood cells, but they can't go on to become nerve cells, for example. Diffusion. Diffusion is the net movement of particles from a high concentration to a low concentration down a concentration gradient. Because it's down a concentration gradient, think of it a bit like a ball rolling down a hill, it's passive and you don't need any energy to do it. Some examples are oxygen diffusing into your cells and carbon dioxide diffusing out of your cells. The bigger the gradient, the faster the diffusion. Higher temperatures also speed up diffusion because the particles are moving about much faster and are more likely to bang into each other. That's a nice link to kinetic energy in physics. We can't talk about diffusion without referring to the special type of diffusion in liquids, osmosis. So osmosis is the movement of water across a partially permeable membrane. So that membrane is partly permeable. It allows some things through and it doesn't allow other things through. Think about the organelles that we just identified. So this means that particles will move from a dilute solution to a more concentrated one. And this often trips up students. Dilute solution, does that have a lot of water or a little bit of water? If you think about maybe a glass of Ribena, if it's dilute, you add lots of water to it. If it's concentrated, it doesn't have much water in it. So osmosis is where we move from a dilute solution with lots of water to a concentrated solution which doesn't have lots of water. Again, it's down a concentration gradient, and so it does not require energy. We have different types of solutions and special words to describe them. Hypotonic solutions. A hypotonic solution has a higher concentration of water outside the cell than inside the cell. So water moves into the cell by osmosis down a concentration gradient. In plant cells, the vacuole swells and pushes against the cell wall, and this makes the cell turgid. This is not a bad thing. It helps support the plant and keeps it upright. In animal cells, water also moves into the cell, but because animal cells don't have a cell wall, they can swell and burst. Isotonic solutions. In an isotonic solution, the concentration of water is similar inside and outside the cell. So water moves in and out at the same rate, and there's no net movement of water in any direction. Animal cells remain normal in shape, but plant cells become what we call flaccid, which is a little bit wrinkly because there's not enough pressure against the cell wall to make them turgid. To try and remember isotonic, remember this: an isotonic drink has a similar concentration to that of your blood. A hypertonic solution. A hypertonic solution has a lower concentration of water outside the cell than inside the cell. Water moves out of the cell by osmosis. In plant cells, the membrane pulls away from the cell wall and the cell becomes plasmolysed. This can cause plants to wilt. In animal cells, the cell shrinks because water leaves the cell. So wilting, think of hypertonic solutions. The way to remember this is if you're sitting in a bath which has lots of water, you end up getting wrinkly fingers. Active transport moves particles against the concentration gradient, this time from a low to a high concentration. This takes energy. The clue is in the name, active. So it requires ATP from respiration. And remember, respiration happens in the mitochondria. The classic example is root hair cells absorbing minerals from the soil. Plants need mineral ions such as nitrates and magnesium for healthy growth. For example, magnesium is needed for chlorophyll. Without it, the leaves of a plant look a bit yellow. At the root hair cells, there is a lower concentration of these mineral ions in the soil than inside the root hair cell already. The plant needs to move mineral ions from a lower concentration in the soil to a higher concentration inside the cell, and this is against the concentration gradient. The plant uses active transport, and this requires energy from respiration to move the mineral ions into the root hair cell. Cells in the small intestine absorb glucose from digested food into the bloodstream. There is a lower concentration of glucose in the small intestine than in the blood already. This means glucose needs to move from a lower concentration to a higher concentration. The cells therefore use active transport, which requires energy from respiration to move glucose into the blood. When we look at transport surfaces, we need to refer to surface area to volume. Smaller objects have a larger surface area to volume ratio, and that means substances can diffuse in and out fast enough. So if you have a very small object and it's respiring, it can easily get oxygen into it. The oxygen does not have far to travel. However, as organisms get bigger, that ratio drops. So they need specialized exchange surfaces. And that might be lungs for gas exchange, gills in fish, or the small intestine for digestion. All of them have huge surface areas. They also have thin walls and a good blood supply to carry gases. A quick exam tip: if you're asked to calculate, work out the total surface area, then the volume, and then divide. Levels of organization. There are 5 levels. In order, cells are the smallest or most basic unit. You don't need to talk about organelles. Cells of the same type group into tissues, for example, like muscle tissue, and you must remember it's cells of the same type. Tissues group into organs, such as your heart. Organs group into organ systems, like the circulatory system. And organ systems together form the organism. If you put tissues before cells, you lose the mark, and it's easy to remember if you go from the smallest to the biggest. Digestive enzymes. There are 3 digestive enzymes that you need to know. Amylase breaks down starch into sugars, and that's why, for example, if you have a piece of bread, you put it on your tongue and you don't chew it, it will eventually start tasting sweet because the starch in the bread starts breaking down into sugar, and that's because of the amylase in your saliva. Amylase is made in the salivary glands and also the pancreas. The next enzyme you need to know is protease, and you'll notice that enzymes have ase, A-S-E, at the end of them. Protease breaks down proteins into amino acids. One type of protease called pepsin is found in the stomach. Protease enzymes are also produced in the pancreas and the small intestine. And finally, lipase. This breaks down lipids into fatty acids and glycerol. Lipase is produced in the pancreas and small intestine as well. The pancreas is a really important organ because it produces several digestive enzymes. You need to know what the enzymes do and what they break down their target nutrient into. Bile is from the liver, but be careful. Bile is not an enzyme. It just emulsifies fats, which means it breaks them up into smaller droplets. It's sort of a helper for lipase so that lipase can work faster. The heart and blood. The heart has 4 chambers. The right side handles deoxygenated blood. The right atrium receives it from the body. Then the right ventricle pumps it to the lungs through the pulmonary artery. So essentially, after you've used up the oxygen in your body, it goes to the right side of the heart. It doesn't have very far to travel, and so just think about the wall of the right ventricle not being very thick. However, the left side handles oxygenated blood, and all that good stuff needs to get around the whole of your body. So therefore, the left side of your heart has to have thick, tough muscle. The left atrium receives it from the lungs through the pulmonary vein, and then the left ventricle pumps the blood around the body through the major artery called the aorta. Valves stop blood flowing the wrong way, and you find them in veins where blood is flowing under low pressure. Because it's under low pressure, there's a danger of it going backwards. So these valves stop blood flowing backwards, keep it going in the right direction. Humans have a double circulatory system because blood passes through the heart twice in one complete journey around the body. One loop carries blood to the lungs and the other carries blood to the rest of the body. And I always remember arteries away. So arteries travel away from the heart and veins travel towards the heart. Plant tissues. A leaf is built for photosynthesis. The palisade mesophyll near the top is packed with chloroplasts. The spongy mesophyll below has air gaps for gas exchange. You then have stomata on the underside, and these open and close controlled by guard cells that let carbon dioxide in and oxygen out. There are 2 transport tissues, xylem, which moves water and minerals up from the roots, and phloem moves sugar in both directions between sources and sinks, and we call this translocation. Don't mix them up. I remember phloem for food, so that's for the sugars moving in both directions. Okay, pathogens. There are 4 types of pathogens that cause communicable disease. So those are diseases that you can catch. A pathogen is a microorganism that causes harm. Of course, not all microorganisms cause harm. If you think about good bacteria, they are not pathogens. And a communicable disease is a disease that you can catch or become infected. Bacteria like salmonella that give you food poisoning are an example of pathogens. And viruses that cause measles or HIV. Fungi like rose black spot on plants. So plants can also be infected by pathogens and get diseases. And protists like the malaria parasite that's carried by mosquitoes. I have seen students writing that mosquitoes are pathogens. Nope, they carry the malarial parasite. And it's a protist. In the exam, you'll often need to give one example of each type of pathogen and the disease that it causes. But your body is not defenseless. We have many different ways that we can try to prevent infection. So before the immune system kicks in, your body has what we call non-specific defenses. So the immune system is specific, but we also have non-specific defenses. Firstly, the skin is a physical barrier, and it's quite good at not letting things through. Obviously, if you have a cut in your skin or a tear in your skin, there is a danger of a pathogen entering and causing infection. We also have mucus and tiny hairs called cilia in our airways to trap pathogens. If you think about the tiny hairs in your nose, they can also filter pathogens and try to prevent them from entering your respiratory system. Stomach acid is pretty good at killing most pathogens that you swallow. Tears contain an enzyme called lysozyme, and that destroys bacteria. And when you cut yourself, platelets clot the blood and form a scab to seal the wound to try and prevent infection. So these are non-specific and the first lines of defense. They don't target specific pathogens. However, we also have an immune response. White blood cells defend you in 2 ways. Phagocytes engulf and destroy pathogens. That process is called phagocytosis. And it's to do basically with eating pathogens, a bit like Pac-Man. Lymphocytes do something more clever. They recognize the unique antigens on the surface of a pathogen, and they produce antibodies that are specific to these antigens. The antibodies bind to the pathogen and they mark it for destruction, which might be a phagocyte coming along and eating it. Memory cells are lymphocytes that hang around in the blood after the infection. So if the same pathogen comes back, you respond much faster. As a common error that students make in their exam, antibodies don't actually kill pathogens, they bind and mark them. Vaccination. Vaccination uses dead or weakened pathogens, and there are 6 steps. First of all, you get the vaccine, often by injection. Secondly, lymphocytes recognize the antigens in that specific immune response. Next, they make antibodies. Step 4, those memory cells stay in the bloodstream long term. Step 5 is if you ever meet that pathogen again and become reinfected. And finally, step 6, the secondary response is fast and strong. So you likely don't get ill. If enough people are vaccinated, the pathogen can't spread easily, even to unvaccinated people, and that is called herd immunity. Photosynthesis. Now, this is almost guaranteed to come up in paper 1, so make sure that you know it and can write it perfectly. So the word equation for photosynthesis is carbon dioxide plus water in the presence of light energy, so we write it above the arrow, and also chlorophyll, so we write that above the arrow, make glucose plus oxygen. So photosynthesis will ultimately make food in the form of glucose, and also, very lucky for us, photosynthesis makes oxygen. This is why people do say have a plant in your bedroom, because they believe it helps to produce more oxygen for you. The carbon dioxide enters through the stomata. Water comes up through the roots via the xylem. The light energy is captured by chlorophyll pigments in chloroplasts. The glucose is used by the plant for energy or to make starch. Think of a potato, for example. The oxygen is released as a waste product. Remember, the reactants are on the left and the products are on the right. Limiting factors. The rate of photosynthesis is affected by 3 main factors: light intensity, carbon dioxide concentration, and temperature. Whichever is in shortest supply limits the rate. So if you increase light, but the plant is short on carbon dioxide, then the rate will be limited and we'll see that flat line, which we call a plateau. Above about 45 degrees, enzymes start to denature and the rate drops sharply. So temperature has an optimum, light and carbon dioxide just plateau. Respiration. There are 3 types that you need to know about. Aerobic respiration uses oxygen. Glucose plus oxygen makes carbon dioxide plus water plus a lot of energy, and it happens in the mitochondria. You'll notice that the equation for respiration is the reverse of the equation for photosynthesis. Anaerobic respiration happens in muscle cells during hard exercise when oxygen runs low. There's not enough oxygen to go around your body to meet the demands of your cells. So instead, glucose breaks down, but not fully. We get lactic acid and a little bit of energy. This is where you might feel a burn or a cramp in your muscles. Anaerobic respiration in plants and yeast cells is called fermentation. Glucose makes ethanol plus carbon dioxide. Because there isn't enough oxygen to break the glucose down fully into carbon dioxide and water, we get ethanol and carbon dioxide instead. That's how we make bread. The carbon dioxide makes the dough rise, and this is how we make beer. So ethanol is a type of alcohol, could be beer, could be wine, it could be spirits, and it really depends on what the plant is in the first place. So the products depend on what's respiring. Mussels give lactic acid, yeast gives ethanol and carbon dioxide. And try not to mix them up. Another common issue is that students will forget to put carbon dioxide. It just disappears. But think about how important it is in making dough, for example. Finally, exercise. When you exercise, your muscles need more oxygen. three things happen. Your breathing rate goes up, your breathing depth goes up, and your heart rate also goes up. And this is all to deliver more oxygenated blood to the muscles. When you're exercising heavily, demand for oxygen outstrips the supply of oxygen. And this is when your muscles switch to anaerobic respiration and lactic acid will start to build up. When you finish exercising, you keep breathing hard. That extra oxygen breaks down the lactic acid, and we call that the oxygen debt. Sometimes you'll see it referred to as EPOC, EPOC, excess post-exercise oxygen consumption, a bit of a mouthful. And that's it. Good luck in your exam.